Most people ask us why we choose glioblastomas first. Actually, I think the question should be, why wouldn't we choose glioblastoma first? Glioblastoma represents one of the clearest examples of disease where current therapies struggle. It is very invasive, it's highly heterogeneous, and it is profoundly immunosuppressive. Once the disease recurs, treatment options usually become really very limited. So if our platform truly has the ability to deliver a localized chemotherapy and localized immune function, then GBM would exactly be the type of disease that should matter.
Engineering a cell to carry two payloads is one thing. Making it find a tumor, do its work, and stop before harming anything else is another challenge entirely. In this second part, Jun Yung Woo takes us deeper into AGEM Bio's approach, from the mechanisms behind their lead candidate AGEM 102 to the questions of control, persistence, and manufacturing that decide whether a promising platform becomes a real therapy. All of it pointing toward a potential new source of hope against glioblastoma, one of the most savage cancers in medicine. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. In your phase 1 study, you chose a challenging target, chose glioblastoma, which is one of the most aggressive cancers. Why going after such a difficult target?
Most people ask us why we choose glioblastomas first. Actually, I think the question should be, why wouldn't we choose glioblastoma first? Glioblastoma represents one of the clearest examples of disease where current therapies struggle. It is very invasive, it's highly heterogeneous, and it is profoundly immunosuppressive. Once the disease recurs, treatment options usually become really very limited. So if our platform truly has the ability to deliver a localized chemotherapy and localized immune function, then GBM would exactly be the type of disease that should matter. And what also intrigued us is really the biology itself. Many of these GBMs actually have defects, innate immune signaling. Some of them have sting silencing. Others have deletion in chromosomal regions, for example, the 9p21 locus. In the entire interferon cluster is deleted. In other words, these tumors have been maybe particularly deficient in the pathways that we are trying to target. So we did not choose GBM because it was easy. We chose it because the biology was really compelling and we really think that we can make a difference in the treatment of GBM.
And how does the treatment look like from a patient's perspective? And also, what are the challenges? Because you have the blood-brain barrier, for instance. How do you deliver whatever you need to deliver in the brain? How does that work?
From a patient's perspective, everything begins with surgery. As you may know, even in a recurrent case, standard of care is surgery. So you have to do surgery to do the debulking of the tumor. So for these recurrent patients, if they are eligible for a second surgery because they have already undergone a first surgery which failed, if they are eligible and they consent to participate in the clinical trial, then AGEM 102 would be administered during the surgical resection. So the cells will be prepared before the surgery and they'll be delivered directly to the resection cavity after the tumor is removed. So from our perspective, this is one of the best approaches. The surgeon has exposed the area and the residual disease, most likely there will be residual disease because in brain surgery, you cannot resect a huge margin. And this is exactly where we want to place these MSCs to chase that residual disease. And after the surgery, after the patient has been given the cells, patient would then take oral 5FC and the engineered cells will then act as local bioreactors to convert the 5FC prodrug into the active 5FU chemotherapeutic drug within the surgical bed. The primary goal of this phase 1 study is really safety, but naturally also we will be looking for other signals of biological activity, progression-free survival, so on and so forth. So for a disease like GBM, where the survival is really very low, even some modest improvements could be very meaningful for these patients.
Absolutely. I mean, that's a huge therapeutic need, no doubt about that. How do you handle with the diversity of that cancer? You have all kinds of different pathways. How does that work? How can you treat a huge variety of different patients?
GBMs, right? They are really two defining characteristics of GBM. And it also happens to be the two reasons why we believe that MSCs can really be effective in this instance. So the first one is GBM is really very infiltrative. Even after what seems to be complete surgical removal, there's always microscopic lesions that remain. And that is one of the reasons actually why recurrence is almost inevitable in a lot of these patients. So we try and overcome this by placing MSCs directly into the surgical cavity so they can chase this residual disease. The second, I think, as you were mentioning just now, is heterogeneity. GBMs are notoriously heterogeneous. And this is where our strategy really differs from a lot of the antigen-directed approaches, such as using immune cells, CAR Ts, and so on. So we are not relying on specific recognition of a specific tumor antigen, but instead we are targeting the biological vulnerabilities that are broadly relevant across the different tumor subclones. Like I was mentioning earlier, the DNA damage, immune suppression, interferon deficiency. So these are really common characteristics or common things that the GBM cells in general, they would possess. These really a lot of the shared hallmarks that a wide variety of the cells will have. And that is why we believe that this approach has the potential to really remain effective even within such a heterogeneous tumor population.
With your allogeneic therapy, how do you overcome immune rejection?
One of the main reasons why MSCs become so attractive clinically is because they have a very unique immunological profile. Unlike many of the other cell types, MSCs express relatively low MHC molecules. So these are involved in immune recognition. And apart from that, the MSCs also secrete a lot of factors that actively modulate immune responses. So as a result, the allogeneic MSCs that we deliver, they have been used extensively in a wide variety of clinical trials, and they have a very strong overall safety record. So approximately 50% of all MSC trials worldwide actually use allogenic sources. What's interesting is that the tumors themselves actually often contain MSC-like stromal populations. In some ways, the tumor microenvironment is already accustomed to interacting with cells from this lineage. So that doesn't mean that immune rejection is impossible, but it is probably the case that MSCs will provide a uniquely favorable starting point to developing off-the-shelf cellular therapies.
Assuming your clinical trials are successful, at one point you will need more material and hopefully will be able to treat more patients. This directly correlates with bigger volumes. And well, on one hand, you have a huge advantage because it's an allogenic therapy. So I think scaling is potentially easier than with an autologous therapy. On the other hand, you have quite some challenges because it's donor dependent. You have variability there. You have variability also with respect to the manufacturing process itself. How does the whole scale up in GMP production work for this once it will go into a commercial production?
When we present ourselves as a modified MSC therapy, a lot of people think that modification is the hard part. Well, I think that the hardest part of challenge actually isn't so much the transfection. The hardest challenge is really the reproducibility of this process. Anybody, they can make a great batch once, but can you make a great batch hundreds of times under GMP conditions? And as you rightly pointed out, every donor is slightly different. every expansion behaves slightly differently. Every manufacturing step really introduces a lot of variability. So the challenge is creating a process that is robust enough that the final product actually remains functionally equivalent despite some of these variabilities. One thing that we are particularly proud of is that we have designed the engineering platform with all of this translation in mind since the very beginning. And many technologies work beautifully when you on the bench scale, but it's a nightmare when you want to scale in GMP. So we've actually spent years simplifying the workflow, defining critical process parameters, and developing a lot of the potency assays to ensure that the technology transfer is smooth. So for us, I think we do have a process in place, and this process, we have iterated it so many times that we know that it works very well in a GMP setting. And along the way, we have developed a lot of know-how to tune this process. So I think that's what makes this process so robust and reproducible at a GMP scale.
This is fantastic, and I'd like to commend you for starting with the end in mind. And this is a message I have been stressing often here on this podcast. So Smart Biotech Scientists, take this to heart when you're developing a new therapy. Think about GMP manufacturing from the very beginning. So thank you so much, Jung, for pointing this out. This is exactly the way to go. So let's talk about the potential of this therapy. And currently you're focusing on glioblastoma. Beyond that, do you have other indications in mind? And eventually, where do you see your platform evolving to?
I think honestly, GBM, glioblastomas, they are just the beginning. So when we think about AGEM 102, we don't think of AGEM 102 as a glioblastoma therapy, right? We think about it as platform therapy, a cancer therapy. The real question here is whether these MSCs or whether engineered MSCs can become a generalizable delivery technology. And if the answer is yes, if the answer is AGEM 102 can be a generalizable delivery technology, then the possibilities are really quite broad in solid tumors, in sarcomas, liver cancers, peritoneal malignancies. These are just some of the cancers which we have worked on in preclinical studies, and potentially even diseases outside of oncology, for regenerative medicine, for chronic inflammatory disorders. So the exciting part is that once we have solved the engineering problems, the manufacturing problems, changing the payload becomes very easy, and the cells remain the same, the MSCs remain the same, but it's the biology that you encode into these cells that become different. And that is what we hope to develop. And that is really the essence of MSC 2.0.
As we are wrapping up, what additional questions should I have asked?
I think one thing that we get quite a lot of questions about is after the phase 1 clinical trials, how do we plan to bring this into further trials and so on and so forth? Or what is the next step for AGEM Bio? I think coming from Singapore, we understand that Singapore is a small country. It's a great country, great healthcare infrastructure, but it's a small country and the patient population is relatively small. So even for the glioblastoma trial, for example, we are thinking of 10 to 12 patients, and our clinicians inform us that, 10 to 12 patients maybe may take 1 to 2 years to do the recruitment and so on and so forth. If we want to bring this further, we would definitely need to look overseas, to look for partners, to see how we can collaborate to bring this therapy to even more patients. When thinking about developing a biotech or thinking about developing such a therapy, I think an important question is to think about beyond the current, beyond the now. What do we envision for this therapy? If it's successful, or even if it's not successful, what are we going to do? So I think for AGEM and AGEM 102 in particular, we know that there is this limitation, and we are actively looking for overseas partners, clinicians, cancer centers who may be interested in working to collaborate to bring this to even a larger patient pool.
What is the most important takeaway from our conversation, Jung?
I think if there's one thing I'd like people to remember, it is this thing. For a very long time, we have treated stem cells as the therapy. I think the next chapter is treating stem cells as programmable therapeutic vehicles. Once you can reliably engineer them, manufacture them, you can preserve their function, you are no longer limited to what the cell naturally does. You are limited to what biology you can encode into these cells. So that is the vision behind MSC 2.0. And I think for us, we are only beginning to scratch the surface of what is possible with these cells.
Fantastic, Jung. Where can people get a hold of you and learn more about MSC 2.0?
You can look me up on LinkedIn, Jun Yung Woo. I'm on LinkedIn. You can visit our website, agem.bio, and you can reach me directly via email, yung@agem.bio, yung@agem.bio. Thank you.
Excellent. Thank you so much, Jung, for being on the show today, for sharing your passion and also providing a solution for a therapeutic area where there is little hope at the moment, but we do need people like you to make a way. And Smart Biotech Scientists, you have all the notes and the infos in the show notes. Please reach out to Jung and his team. And Jung, thank you once again for being on the show today.
Thank you so much, David. Definitely a pleasure talking to you.
The story of engineered MSCs is at its core a story about precision, directing a cell to the right place, controlling what it does once it arrives, and proving that what works at the bench can be made reliably at scale. If this episode was worth your time, please leave a review on Apple Podcasts or your favorite platform. It helps others find the show. Thank you so much for tuning in, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.